Example here is the virus against which resistance is developed is CMV, cucumber mosaic virus, and tobacco mosaic virus, species modified Nicotiana tobacum. However, the source for antisense RNA is the CMV and TMV. Transgenic plants are also able to develop resistance against fungus. Fungi are the organisms which have major role in decomposition of organic matter. Strategies here to provide fungal resistance is damaging the cell wall or producing defense related proteins against fungus. First is the phytoalexins. Phytoalexins are the secondary metabolites which are produced by the plants in response to the fungal infections. Example of the phytoalexins are terpenoids, glycosteroids, alkaloids. These have a broad spectrum of antimicrobial activity. These phytoalexins have the fungistatic effect by puncturing the cell wall and delay the maturation or disrupt the metabolism or prevent reproduction of the pathogen. By these multiple effects, it is able to have the fungistatic action. So, the strategy here is to introduce the gene from the other transgenic gene source like peanut stilbene into the Nicotiana tobacum, which may lead to the formation of elicitors. Uh, elicitors may be due to the elicitors produced by the host and fungal cell breakdown or by abiotic agents such as mechanical injury, ultraviolet radiation, and heavy metals. The product formed after in introduction of gene is the resveratrol. Next strategy is to induce the formation of an enzyme called chitinase. Chitinase is an enzyme which damages the cell wall of the fungus by breaking the glycoside bonds between the glycosidic bonds between the cell walls of the fungus. Chitin is made up of is a here the you can look at the structure of the chitin. This is a glycosidic bond. So chitinase is an enzyme which breaks this glycosidic bond, as a result of which the cell wall of the fungi degrade. Here the gene source is the Serratia maricens, species modified Dicotiana tabacum, and fungal pathogen against which resistance is developed is Alternaria longipes. This is the other example. Next strategy is to form ribosome inhibiting proteins. Ribosome inhibiting proteins, which means that seeds of various cereals contains proteins that are toxic to some fungal pathogens. So these protein inhibit the ribosome function by breaking the glycosidic bond in the ribosomal RNA, as a result of which peptide elongation and consequently the growth of fungi is arrested. So we can develop the resistance of plant towards fungal pathogen. Example here is the barley is the gene source which produces glycosylase enzyme which cleaves a glycosidic bond in the 60th subunit of ribosome. Species modified is the which means transgenic plant developed is Nicotiana tabacum. Fungal pathogen is Rhizoctala solani. Next, transgenic plants can also help in developing resistance towards stress. Stress is nothing but refers to any change in the environmental conditions that might reduce or adversely change a plant growth or development. Plant depends on internal mechanism for tolerating these variations in the external environment. So the strategy, what is the strategy here? The water deficit stress is taken into consideration. Water, if there is less amount of water in the soil, what are the effects? Drought, heat and cold, and salinity. These are all the effects if there is a water deficit in the soil. When there is water deficit in the soil, there is a deficit between the supply of water from the root and loss of water from the leaf by transpiration. This may lead to dehydration and death. So how to combat this? Transgenic mechanism has provided a solution for this. How? Idea here is to cause the accumulation of solutes like osmoprotectants, which have the property to provide stress tolerance to the plants. How can we provide stress tolerance? The water from the roots is getting transported from the roots to the leaves and then getting transpired by transpiration. So water is getting lost. So the idea here is to restrict the movement or slow down the movement of the water from the roots to the leaves by a formation of different solutes like osmoprotectants. Different osmoprotectants used here are amino acids like proline, quaternary ammonium compounds like glycine, betaine, polyols, and sugars like mannitols. First, we look at the proline. Proline means they have an they are also a phytoosmoprotectant, which means they accumulate in the cells so that water movement from the roots to leaves is reduced or slowed down. The result is enhanced by production accumulation of proline. When you look at the biosynthesis of proline, you can see that there is an enzyme called D-pyrroline 5 carboxylate synthase. D-pyrroline 4 carboxylate synthase. Here, delta pyrroline 5 carboxylate synthase. So, strategy here is to introduce the gene from other source into the plant so that it produces D-pyrroline 4,5 carboxylate synthase enzyme, as a result of which more proline is produced, which acts as an osmoprotectant in combating the stress in combating with the stress. Gene source is from V. aconitifolia into tobacco cultivar xanthe. Gene name is P5CS. Next strategy is to next compound which can be called as osmoprotectant glycine betaine. Here also the biosynthesis of glycine betaine is induced by production of enzymes like choline monooxygenase and betaine aldehyde dehydrogenase. These are the choline monooxygenase and beta. So these are the enzymes involved in the production of glycine betaine. So genes are introduced from other sources into the plant so that these two enzymes are produced more and glycine betaine is also produced. Next strategy, next compound which can be called as osmoprotectant mannitol. Here also the biosynthesis of mannitol is induced by enhanced production of an enzyme called mannitol 1 phosphate dehydrogenase. Here you can see that in the production of mannitol 1 phosphate dehydrogenase. This is the enzyme which is induced by introducing the gene from other sources like bacteria introduced into tobacco plants and resistance to salt stress. So by the production of these protectants, plants are also uh, developing resistance towards stress. So as you can see that transgenic plants are helpful in developing resistance towards herbicides, uh, fungi, virus, insects, and also towards physiological stress. So I think this video might be helpful in giving an overview of the applications of the transgenic plants in different ways.